Okay, let's see what you've got. So the first sentence, in the search for new biofuels, research has been done into the digestion of wood waste by fungi. So the first word that we highlighted there was biofuels. So looking that word up, we found out that it's an energy source that's being derived from biological material. Now, rapeseed oil, the crop that you see that's bright yellow, this is a common one that's being used as a biofuel at the moment, as a replacement for diesel in diesel engines. So if you're interested in biofuels, maybe that's something to look up at another time. Anyway, so research has been done into the digestion of wood waste by fungi. Now, we didn't mention this before, but the word digestion, I think sometimes that's taken for granted, isn't it? You think, eat your food, you take it into your digestive system, it gets broken down, but that's quite a vague description of what digestion actually is. So in terms of an A-level standard answer, if we're talking about digestion, we're talking about the breakdown of larger molecules into smaller soluble molecules, because smaller soluble molecules are able to be taken up by cells, and they can utilise those small molecules then to make the molecules that they need to function. So an example over here, we've been talking about proteins recently in class. So a protein is an example of a large molecule, and proteins can be broken down into their monomers, which are amino acids. So this is an example of a digestive process here. Wood waste. So if we think about what wood waste is, maybe we're talking about things like tree bark from decomposing trees. You also get sawmills, wood factories, and you get waste products there as well, where you could get waste products of a wood nature. Now, fungi, as I said before, this is part of the classification unit in F212, so you'll find out a little bit more about fungi. But basically, they're an organism that have been classified into their own kingdom. You might remember talking about 5 kingdoms at GCSE. It's something that we look at a bit later in F212. And when we do that, you'll find out a little bit more about fungi. So these are the main points that I think you might have found out during some research. So if you think about plants have cell walls that are made of a substance called cellulose, as you know. Fungi don't have cellulose cell walls, they have chitin cell walls. They also have multicellular bodies, so they're made up of many cells, they're not just one cell. And that body of cells is called a mycelium. And they produce these long filaments and these long projections that are called hyphae that protrude into the food source that they're actually living on. They're multinucleate, which means that they contain more than one nucleus within what looks like a cell. And they also produce spores. Like I say, you'll learn a little bit more about fungi once we get to the end of the course. And think about it, this is an enzyme question, but it is drawing upon that classification unit too, and I know we've not studied that just yet. It's not fully relevant here, but it could be. Okay, so the next bit. The fungi produce cellulase enzymes that break down cellulose into sugars. Okay, so like I said before, cellulase, it's not an enzyme specifically that we've named before. It does already tell you what the substrate and the product is for this enzyme. It says it breaks cellulose into sugars. So just to get that really, really clear in my head, I've written over here cellulose being broken down into sugars. Now, cellulose is a large molecule. Sugars are small and soluble. As you know, you put sugar in your tea, it dissolves. So sugars, they're soluble. You should know that already. So this is an example of a digestive process, breaking large molecules into smaller soluble molecules. Okay, cellulase is an enzyme. So all the basic information that you know about enzymes already, you know the globular proteins, you know that the monomers are amino acids, you know they're biological catalysts, they speed up reactions without being used up. And they have a specific 3D shape, and that's due to the tertiary structure. Do you remember proteins have 4 levels of structure, and it's the tertiary level that gives a protein its specific 3D shape. And in terms of an enzyme specifically, it's the active site shape that's really important because it's complementary to the substrate. So remember, they fit together in that lock and key action. Okay, next part of the question then, it says these sugars can then be converted into ethanol. So like I say, unless you do chemistry, you wouldn't know much about ethanol, so we've had to research that. Ethanol, it's a colourless, water-soluble alcohol. You won't know much about alcoholic drinks just yet until you turn 18, but ethanol is the principal alcohol that you find in alcoholic drinks that you can buy in the pub. This is the formula of ethanol, C2H2OH. So like I say, if you do chemistry, you'll learn a little bit more about the bonding between the atoms there. Now it's produced in yeast during a process called fermentation. So when sugar is fermented in yeast, you get this reaction here, glucose being converted into the ethanol along with carbon dioxide. This is actually something that we look at further at A2 level. But the question mentioned ethanol here, so if we didn't know what ethanol was, it's only right that we look it up because we don't know what information is going to be important when we get to the question. Okay, so in terms of the stages of the this digestive process, so we have cellulose being converted into a disaccharide by this enzyme that they've named just enzyme 1. So in terms of cellulose, what that is, so after a bit of research, it's a carbohydrate, it's a polysaccharide. Now we've been looking at proteins and we call those polypeptides. So poly, as you should know, means many. Now cellulose is actually a molecule that's made up of many glucose molecules. And there's two different types of glucose molecules, and the one that we've got present here is beta glucose. You know, every other glucose is actually upside down, and that's what makes cellulose consist of long straight chains, and these chains are held together by hydrogen bonds, and we can see the hydrogen bonds here. Remember, they're always represented by dashed lines. You'll learn a little bit more about cellulose when we actually get onto the carbohydrate topic after proteins. Okay. So cellulose is converted into a disaccharide. Now di, if you remember, means two. So if you see here, cellulose was made up of long chains of these beta-glucose molecules, and it's broken down into just units of two. So remember, di means two, so we've got two beta-glucose molecules here that this enzyme chops the cellulose molecule up into. Okay, then we've got a second enzyme, enzyme 2, and we can see that the disaccharide then is hydrolyzed. It's cut, that bond's broken there, and it's converted into this single unit. And just mentioned over here, this unit, C6H12O6, is actually the formula for glucose, which is a sugar. So at the bottom here, just made it very clear in my head. It's always good to rewrite things, put things in a bit of a simpler format to try and get it really, really clear in your head what information has been given to you. So enzyme 1 converts cellulose, which is a polysaccharide, into disaccharide. So I know that the substrate for enzyme 1 is cellulose, the product of that reaction is a disaccharide. And again down here for enzyme 2, converts a disaccharide into glucose. So the substrate for enzyme 2 is a disaccharide, the product is glucose. And we had this question earlier, how come we've got two enzymes? So if you remember, enzymes are specific. They have a specific shaped active site. It's only complementary to one substrate. So if you look, we've got two different substrates here. Cellulose was the substrate for enzyme 1. So enzyme 1 must have an active site that's complementary to cellulose. Okay. Enzyme 2 then doesn't have an active site complementary to cellulose. It has an active site complementary to disaccharides instead, so it's not able to break down the cellulose. It can only break down the disaccharide. So we need two different enzymes, one for each reaction, because the two enzymes have different active sites. So if you remember, enzymes are specific, they show specificity because of that. Okay, so Obviously, in an exam situation, you haven't got an A3 piece of paper, you haven't got all of this space to write everything that we've just written just now. This is just practice. We're trying to train you into how to think. So all of these things that we've picked out, obviously by the time you get to your exam at the end of May, beginning of June, all of this will become second nature to you. But this is how you should be thinking. You should be looking at these words that are being used, biofuels, digestion, wood waste, and as you're reading, all of this stuff should be firing in your head like little fireworks going off. You should already be building up a little bit of a diagram like this in your head with all this information before you get to the actual question. If you do this, these sorts of questions, these application questions will become much easier and a little bit less daunting to have a go at.